Hi, my name is Alyssa D'Aiuto, and I am a Sustainable Food Systems major at the University of Delaware, studying the Bacillus valenciensis strain S4 bacteria as part of the UD Envision undergraduate research program. My research takes place in Warlow Hall, which is one of the primary laboratory buildings for students and faculty of the College of Agricultural and Natural Resources at the University of Delaware. As we all know, our global population is continuously growing, as is the need to increase food production to feed everyone. Plant diseases lead to a reduction in crop yield, which can be detrimental to our goal of feeding the world. This bacteria has the potential to be a biological way to fight fungal plant pathogens. The overarching goal of this research is to assess if the bacteria will inhibit the growth of various fungal plant pathogens and create physiological improvements in tomato plants. To do this, I have two main focus areas, the first being explant assays, where I studied the microbial interactions between the bacterium against different fungi found amongst UD botanic gardens. I hypothesized that the bacterium would cause at least 30% inhibition in the explants. I conducted a series of tests to determine if the selected pathogen is susceptible to the bacteria by growing them in the vicinity of each other. Within the 6 different explants, I found various different levels of inhibition. The fava bean explant and the crab apple mummy both had the highest percentage of inhibition with over 50%. Moving on to the second area of focus, which were my tomato experiments. During these, I wanted to study the physiological differences in tomatoes when treated with S4 compared to ones that weren't. I hypothesized that the bacterium would increase root growth compared to non-inoculated plants. To test this hypothesis, I applied S4 root drenches to 1-week-old sprouts and added a boost 28 days later. S4 was applied as a seed treatment, rotated in it for 5.5 hours, then planted to measure germination rates. And most recently, germination assays were set up with S4 treated seeds to measure different germination traits. As of right now, there are no physiological differences that are noticed. We are still waiting for fruit to develop on the root-drenched plants to pull up the roots and examine them. S4 treated seeds only had 1 additional germinated seed compared to the control, and we are still awaiting the results from our first germination assay. Some of my goals moving forward are to test various parameters in the germination assays, use different tools to test physiological traits of the tomato plants, such as photosynthesis rate, and to continue to test more pathogens against S4. Within that, I think it could be beneficial to use the supername of the bacterium culture to test for inhibition, as this could save time and resources. This bacteria has continually proven to have the potential to be a biological control to various plant pathogens, especially ones found in Delaware. The use of this bacteria in the field can help to promote more sustainable agriculture by reducing the use of fungicides and other chemicals.